her today in our series. So Lisa received her PhD in molecular and cellular biology from the University of Harvard in 1997. And during her PhD, she used a molecular genetic approach to study the mouse behavior. After that, she joined the laboratory of Dr. Catherine Dulac and to carry on her postdoctoral studies. And after that, Stowers started her lab and became an associate professor at Scripps Research in 2002. So today, she serves as associate professor in the Department of Molecular and Cellular Neuroscience at the Scripps Research Institute, and her research on how neurons sense and respond to pheromones, especially the molecular pathways in mouse behaviors. She has made enormous discoveries connecting pheromones and behavioral responses in mice, including scent compounds that lead to interspecies fear, male-to-male aggression, pup suckling, female reproductive behavior, and scent making. Very happy to be hosting Lisa today, and I look forward to hear from her. So thank you so much for joining us, Lisa.
Thank you, Zita, for the nice introduction. It's a pleasure to be here. My sound is kind of going in and out a little bit, and I lost Zita a couple times there, so hopefully we'll be able to stay on throughout the talk. Please let me know in the chat or let me know, Zita, if there's a problem. Today, I'd like to share with you some of the work that we've been doing in my lab. My lab's interested in understanding how external stimuli drives behavior. How does an animal sense the external world and make a computation to do an appropriate action? But as we're coming to realize, the animal's internal state is also very important for this to happen. In order to get an appropriate behavior, all individuals will act differently given the same environment in general. So this could be due to different experiences that they've had, whether they're in a different developmental state, or, as I'm going to talk to you more about today, whether their internal state is slightly different. And several years ago, we started looking at this when a postdoc in my lab wanted to understand the differences between male and female behavior. Female behavior is a great model for internal state because their behavior isn't always static. So a female is robustly attracted to a male, but not all the time. And as you all know, that varies with her ovulatory cycle. And in the mouse, the ovulatory cycle is called the estrus cycle, and it's very rapid in our lab strains. So the whole cycle is 4 to 6 days in our lab strains, where she's behaviorally receptive towards mating when she's in estrus. And we can show that with this odor assay here, where we take a male odor and we place it on one side of this arena here and a control odor on the other side, and just watch her behavior, her locomotion, and see where she spends more time. And as you can see by this heat map of where she's traveled, with the warmer colors indicated longer presence, that she spent more time on this side of the cage with the male odor when she's in estrus. And that's an indication of her behavior. Not a very high resolution indication, but it's some indication. And if we take the exact same female and just wait a couple days and do the same assay with her, we find that she spends much less time in the vicinity of the male odor and more time being sort of indifferent or even on the opposite side of the cage. So we were really excited to be able to use this model where we're not comparing two different groups of individuals, but really the same individual as it's in a different internal state, to understand how the brain is changing. And over the course of several years, we had identified some of the chemical signals that are in this male odor. And one of them is represented by these gray blobs here. And those are male cues that are attractive to the female. And so we imagined that the female was really focusing and paying attention to those cues when she was in estrus, and not so much when she was in diestrus. And we were all ready to embark on studying how the brain was changing during this 4-day estrus period to make these different decisions about her behavior. But my postdoc was very interested in first just doing a control to make sure that nothing was changing in the sensory system in the nose. And this to me seemed like a necessary control, because as we know, the sensory systems like the visual, auditory, and here in the nose, they're responsible for gathering data. And there'd be no real logic for it to be attenuated and gather different data during different parts of as the internal state changes. The more data coming in, the better. And then the brain decides what to do with it. And we know this is to be the case with our human behavior. And one great example of this is an auditory response called the cocktail party phenomenon. So back in the days before COVID, when we used to all gather in a crowded room, you may be able to, the room may be very noisy with lots of folks talking to each other, but you can focus on one person and have a nice conversation just by attending, and sort of everything else sort of recedes into a quiet hum. You don't really notice what other people are saying. But if somebody across the room says your name, you may all of a sudden realize you've heard it. You'll know the direction that it came from. Maybe if you recognize whether it's a male or female, and if you know the participants in the room, you may even know who said it. So a lot of this information was getting in the whole time. And as you know, the brain was just gating what we should attend to. And so really, this was an unnecessary control to do. But it was a very easy control. And so not to stifle innovation, there was no way I was going to stop her. So she went ahead. And the way she tested the sensory system was very easy. We postulated that the changes were primarily due to changes in ovarian hormones that change throughout the estrus cycle. And so when you're in estrus, the female has a surge of estrogen. And just a couple days later in diestrus, the estrogen recedes, but progesterone surges. And so she could obliterate these surges by removing the ovaries and just removing the neurons from the nose ex vivo and placing them in a dish for acute calcium imaging. She could add exogenous hormones at physiological concentrations and ask whether the sensory response changed. And this old assay, I still like it very much, so I'm going to show you the raw data here. So these are calcium imaging traces from 4 or 5 different neurons. And as we perfuse on the male attractive pheromone here, you can see that these neurons are all responding. They do it quite robustly several times here. But now when she adds the physiological concentration of progesterone here, and just waits a short period of time, 10 to 15 minutes, and then perfuses on the stimulus, you can see that these neurons do not react at all. They're just completely silent. And because they're in a dish, we wanted to ensure that they weren't just dying as they were sitting here. And so she could add this agonist that gates the channel directly. And sure enough, all the cells are alive. They're able to fire just fine. And so this was our first indication that something was being modulated in the nose in response to the internal state. And this assay is really interesting to us because, first of all, it tells us that the action is occurring in cysts right on these sensory neurons, because these neurons were excised from the brain. It was in a calcium imaging dish, remember? And so this says that the neurons themselves were sensing progesterone. They weren't just receiving a neurological signal from the brain. And second of all, most steroid hormones were known to act as transcription factors. And by changing the expression of genes in the cells, then they change function and behavior. But this was happening too fast for that to occur. It was happening quite quickly, suggesting it was more of a rapid action mechanism. And so what we knew at the time about pheromone sensation was that the ability to detect a wide variety of signals is due to this large family of G protein coupled receptors. There's about 350 of them that are each tuned to a different ligand, but they all thought to plug into a G protein coupled cascade through a phospholipase to gate this trip channel. And that's what's happening here and here. And what Sandeepa found was that these cells also express a hormone receptor that's able to detect progesterone. This is a non-canonical progesterone receptor that has a single transmembrane domain that initiates rapidly a kinase cascade when it binds progesterone. And so in the presence of progesterone, it causes this enzyme, PLC beta 2 here, to be phosphorylated. And when it's phosphorylated, it turns it off. so that the signal transduction can't proceed to gate the channel. And the signal, the neurons never fire. They never tell the brain that the pheromone is out there. And so all in all, what she found was that during estrus, that signal transduction cascade is on, the animal is able to sense the signal, pass it on to the brain, and behavior is able to occur. But during diestrus, when there's a high level of progesterone, it phosphorylates that transduction cascade and basically turns it off. And the female's basically blind to those cues out there. She never senses them, so she never behaves in response to the male signals. So that project, we took all the way down to identifying the residue that was phosphorylated, because to me it was just such an odd event to have the female not detect these sensory cues. And I got a lot of, this was several years ago, I got a lot of guff, mostly from my male colleagues, saying that, well, you know, this makes perfect evolutionary sense. Why should the female sense the male if she's not receptive, physiologically receptive for mating? She'll just be so distracted by these male cues, she'll, you know, never get anything done. And I thought that was rather ridiculous. Oftentimes in our human behavior, and in animals' natural behavior, there's a lot of sensory cues that are being processed that don't require immediate action, but it's helpful for the animal to know that they're there. So my view is more sensation is better for the animal. Another option was that this was specific to the female sensing male pheromones, because there is a animal behavioral response in that if a female mates with a male and he ends up being killed or leaving and another male takes his place, if she has those pups to term, he will kill them and sire his own. But when she smells the other male taking her place early in the pregnancy, she'll automatically self-abort, and that saves her lots of resources. This is called the Bruce effect, and we thought maybe this was somehow particularly linked to the Bruce effect in mice. But the model the hypothesis that we preferred and ended up following was that maybe we discovered this by looking at females, but maybe this is something that makes the VNO special. Maybe there's a broader program of sensory modulation that occurs in the mouse, and that this was just a first indicator of it. So we started looking to see if other aspects of sensation in the VNO were being directly modulated in the sensory epithelium. And the first thing we did, because it was just such a long process to find a behavior and find a ligand and then understand how that was being modulated in the female under that particular hormonal state, we decided to not look at particular behavioral events and instead look at the system as a total from a molecular point of view. And so we first focused on the enzyme that was being modulated, this phospholipase. And it turns out that the phospholipase signaling that we looked at, if we looked at mutants that did not express this phospholipase beta 2, she doesn't respond at all to the male cues, as I showed you previously. But if I show her a more complex odor stimuli, here's being male urine, we get about half the response as we do in wild type individuals. And if we have a different family of cues here, this is a cat odor, we find that whether she's expressing PLC beta 2 or not, and also whether she's an estrus or not, doesn't make a difference. She can smell the cat just fine through her vomal-nasal sensory neurons. So that tells us that there's some other signaling element also playing a role in the system. And that was surprising to us because most of the chemosensory and GPCR driven sensory systems are characterized by having a large family of G protein coupled receptors to specifically engage with the external environment. But then they plug into a single signaling module that's made up of the same proteins. And that way they're able to have a relatively similar output signal in the sensory neurons that can compare and weigh the identity and quality of the sensory stimuli. But here, this seems to indicate that it's not just PLC beta 2 that's part of this module, but something else is going on in the VNO. So we decided to address that a little more specifically. And it turns out in the mouse, there are about 17 PLC genes. These are entirely different genes. And if we do an RNA seq on the entire vomal nasal organ shown here in cross-section, just grind the whole thing up, we find that the PLC that was thought to be primarily responsible for signaling and is responsible for signaling to that male pheromone, this beta-2 here, is very lowly expressed, and that there's some other PLCs that are more highly expressed. Now, there's also, when we grind the whole thing up, there's supporting cells and blood vessels, but if we look just in the sensory neurons, we have evidence that the sensory neurons themselves are expressing at least 14 of these different PLC family members, and the PLC beta-2 is only one of them. So if we do single-cell RNA-seq, here we're showing 61 different single-cell neurons. And we find that rather than all the cells expressing beta-2, or some of the cells expressing beta-2 and another one expressing one of these different PLCs, we find that there's many of the different PLC families are being expressed. And if we go ahead and use RNA-scope, I think I cut out the slide, but in RNA-scope, we see evidence for all 14 being expressed. And in this instance, what you can see is that primarily, on average, only one is being prominently expressed per neuron. So that's quantified and shown here. So these are all the sensory neurons, and we find evidence that rather than them all being expressed in every neuron, or just particular variants being expressed, that each neuron is expressing a different subtype. And of the mutants that we've tested so far, which so far have just been from this PLC beta family, just like I showed with you with the beta-2 being necessary for the detection of the male pheromones, here we identified ligands that overlap predominantly with the PLC family that we wish to test, in this case, PLC beta-3, and our ligand here, just for kicks, is called sulfated steroids. It's a ligand known to activate the vomal nasal organ. It doesn't activate just PLC beta-3 neurons. There's some others, but in the mutant, you can see that when we knock out PLC beta-3, the response to the sulfated steroids goes way down, suggesting that indeed PLC beta-3 is necessary for transducing this particular ligand, like PLC beta-2 was necessary for transducing the male pheromone cue. And here we do the same trick, but this time beta-4 overlaps best, or is sensing a predator odor. In this case, we use rat bedding. And you can see here that in the mutant, there's still some other neurons that are expressing other PLCs detecting a bit of it, but generally that signal goes down as well. And so what this is telling us is that not only are these expressed one per neuron, but they seem to be the primary signal transduction element in these cells. So very little work has been done on PLCs. We've been CRISPRing several others from different families to see if different family members that have the same pattern of one PLC per neuron are also responsible for doing the signal transduction in their particular cells. But so far, it looks like that there is a wide variety of signal transduction elements. And so overall, we'll go through and test how many of these PLCs are actually responsible. But what it looks like is that we're getting variation both in the wide variety of G protein coupled receptors that are able to be expressed, as well as in the signal transduction pathway that's used to transmit this signal to the brain. So we've been very curious asking why. It's been a very difficult answer for us to address, but we're working on it using the model that we used for the female response during estrus and diestrus. And that is that not all the PLCs these aren't just slightly different variations of different isoforms, but these PLC family members are entirely different genes. They have different functions, and they interact differently with different signaling elements in the cell. Moreover, I showed you that the PLC beta 2 here during estrus and diestrus was silenced as it was phosphorylated. And of the PLCs that have been previously studied for other reasons, they're mostly known to be modulated by phosphorylation. Some of them are silenced. And in these, if they're phosphorylated on different residues, they can also be activated so that they turn up. And some of them are completely silenced until they actually receive a phosphorylation, and then they're turned on. And so we're very interested in this bidirectional modulation. And it suggests to us that maybe they're able to modulate the response to the to others, to more personalize the response to the external world. And so in order for that to happen, there has to be some organization of the system. If these things were coming on and off, for example, if PLC beta 2 was coming on and off in a wide variety of cells, not just in response to the male cues, then perhaps she wouldn't be able to sense the predator cue when she was in diestrus, and that would have consequences towards her fitness. And so one of the things we're looking at is whether the pattern of expression with these PLCs is random and stochastic, or whether it's determined. And we initially planned to do this just with a quick sequencing experiment. Our PLCs are very lowly expressed, which allows us a high level of, we have to sequence very deep. And so it's very expensive for us to do this. We calculated we needed about 30,000 cells to be able to get something meaningful. And that was sort of beyond my budget at this point. So by in situ, we're looking at patterns of expression. It's very hard. The G proteins are very similar to each other. So it's hard to get a specific in situ probe to look at the relationship between specific genes. And also there's 350 different specific receptors. It's just a lot of in situ. So we broke it down into groups. And the vomeronasal system, here's an image from the Dulac lab that shows some receptor probe families and how diverse they are in these different multicolored sensory neurons here. You can see this is the sensory layer. But if we use a different probe that's to one of the G proteins, it divides it into two subgroups, this green group and this blue group. And they're also known to be divided by different subfamilies of vomeronasal receptors. They detect, the blue group detects primarily mouse odors, while the green group detects primarily predator odors. And they project to slightly different places in their first relay in the brain. So they're functionally different. And so we wanted to ask, are all PLCs expressed, even though they're in one per neuron, are they expressed in the blue group, in the green group, or are they mixed? And what we found was sort of an interesting answer, which is a little bit of both. And so here's a ratio of the blue group shown in the solid bars and the green group showed in the hatch bars. And so these family members have a bias towards being in the green group here. They have more overlap with the green probe. These over here are biased to being with the blue group. And then we have a subset in the middle that shows no preference whatsoever. So they're equally likely to be expressed with either the receptors directing the cat odor or the predator odor. The other interesting patterns that we see are that from animal to animal, so this is a summary of 9 different animals across several sections of each of those animals. And you can see that they're not all expressed at the same level. If so, we would expect them the number of cells in each section to be at around this level here. But in fact, we find some that are highly overrepresented. They're in more cells than we would expect. And these are the highly abundant groups here. We find sort of a middle abundant group here. And then we have the low abundance, of which, again, that PLC beta 2 that detects the male pheromones is shown here. It's very rare. And so this is from animal to animal, in males and females, in juveniles and adults. And so we believe that both the pattern of being biased towards one family or the other and this abundance shows us that it's not just random which cell gets it, that there is actually sort of a plan that it's determined. The other interesting thing that we notice from our sequencing and doing in situs is that it's not just the unusual progesterone receptor, which is called PGRMC1 and shown here, that we see that it's expressed in these neurons, but that we see a wide variety of other hormone receptors. Now, this is a hard one for us to, without directly testing the function of each one, to infer, because a lot of neurons express hormone receptors, and a lot of cells do, just as a housekeeping gene, just to keep them functioning normally. So our criteria for making them interesting is that they had to be expressed in the same pattern of a receptor or as the PLCs, and that some cells have them at good levels and others don't have them at all. And so for all of these hormone receptors, they're in an all or none pattern within the VNO, sort of salt and pepper. Some cells are highly expressing these genes, for example, that are implicated in a response to hunger or stress, and some cells aren't expressing them at all. So we find that very interesting, just like PGRMC1 is expressed in some cells, but not all. And so we're interested in understanding this intersection between the hormone receptors, this variety of signaling responses, and the pheromone-mediated behavior. So what I've told you is that there's a variety of different signaling pathways in the vomeronasal sensory system that's unusual for most sensory systems. And that we think that the relationship between the receptors and which cell is expressing a PLC is genetically determined, that it's not random, and that the primary sensory neurons are also expressing these hormone receptors in an all or none manner, just like the progesterone receptor that I described earlier. So what we're wondering is whether these hormone receptors are acting on this varieties of PLCs to be able to turn on or turn off sensation based on previous experience and internal state. And so just as a quick global way to see this, and here what we matter is, what the summary is, that whether these hormone receptors are signaling and modulating the PLCs, not just in response to the estrus-diestrus state. So what we imagine is that if an animal was to sense a cue and it acted like a regular sensory system, like the regular main olfactory system, like the taste system, like the visual system, that whether 5 individuals would all have the same sensory response, the same sensory receptors would be activated in response to the same external environment. But if in fact the system was made to be modulated, we would imagine that individuals from grossly different states, so here we would have a juvenile, an adult male, females in estrus and diestrus, and a pregnant female, that maybe they would have a different response. And so we went ahead and tested this, and we did this just using a molecular trick where we could get a handle, an antibody handle on cells that had been activated. So just like CFOS is sort of a crude way to molecularly identify activated neurons, here we were using, phosphoribo S6, which is a ribosomal protein that becomes phosphorylated active after neural activation. We could do that to do an IP of activated neurons after we show these different individuals the same stimulus. And this is what we see. So this is a qPCR of the IP from these different individuals. And the individuals themselves are along the x-axis, and the PLC variants are along the y-axis. And the first thing you can notice is just the patterns. Here, I've taken the 3 most impressive differences and eliminated the 2 different females. They're more like the pregnant female. But here you can see there's 3 different patterns, and they're grouping by juveniles, males, and here it's actually diestrus females. And what we see is that when we do the pull down after a complex male odor blend, so this is the one that there was a 50% response in the PLC beta 2 mutants, so it's male urine is the stimulus. You can see that there's some PLC variants that are present in all of them. Here H2 and L1 are abundant, but then there's some that are specific. So here you can see H1, L2, and L2 are highly activated in the males after they detect the male stimulus, but they're not activated at all in the juveniles or the diestrus females. And here you can see in the juveniles, cells expressing beta 3 or XD2 are highly activated in the juveniles, but not in the males. If we go ahead and look at the pheromone receptors, which is the key indicator of differential neuron types, we see the same pattern. So here's all 5, but this is the diestrus female, the adult male, and the juvenile. Sorry for the scramble here. But you can see some pheromone receptors are activated in all of them, whereas others are specific either in the juvenile or in the diestrus female. So the patterns are very similar and suggest that actually different neurons were being activated. So overall, what we see is in this crude generalized experiment is that in contrast to passive sensation, which is what we had presumed the VNO was doing, where all the sensory cues were getting in equally to all individuals, and the brain was deciding what to do with those cues, what we think we're seeing is a system that's giving personalized sensation, that based on your hormone repertoire and those hormone receptors that are expressed on the VNO neurons, that different neurons, since different sensory cues are being passed along to the brain through the active modulation of the PLC family. And so different individuals are actually sensing a different environment and they're responding different to it, and that's reflected in their behavior. Okay, so we started this project because we wanted to study the brain. And we love studying sensory neurons, but we really want to study the brain. And what this has told us, if it holds up, is that maybe some of our models of how the brain is responding to the environment and internal state and generating behavior aren't quite accurate. So our previous model was very simple, that an odor cue would be detected at the periphery and passed along, and as it moves and is processed by the brain's making decisions on its experience, whether it's ever experienced the cue before, whether it liked it or disliked it, whether it's a male or a female, whether it's hungry or tired. And all of these things at some point accumulate into a decision, which either the animal goes towards the cue, away from the cue, or just ignores it altogether. But what I just told you was that there's actually some of these decisions could be being made in different places, not just sort of a linear feed forward, simple progression, but that there could be some modulation that's occurring, decision making that's occurring right before the first relay outside of the brain. And so as we learn more about this will be important to allow us to study the decision making that's going on in the brain. If it indeed is occurring, we'll be able to better control it, and we assume get more robust behavior in the brain. But in the meantime, what it tells us is that we shouldn't pre-guess what each node is doing as we see activity patterns change in the brain. That it really is more of a molecular decision and a computation rather than a guess at function. And so people have been studying different nodes that are transmitting that are causing this behavioral decision in response to pheromones for many years. And in my opinion, we still don't understand what the primary nodes are doing. We know how information is flowing sort of, but not fully, and how behavior is being generated in response to odor cues. We don't understand much at all. And part of the reason is because people sort of pick these different regions. The information is traveling primarily in the limbic system through the amygdala and the hypothalamus. And people pick their favorite region, the amygdala. It's doing emotional processing, and then study that without understanding the whole system. And so people like what the amygdala is saying, it's just involved in emotional processing, are seeing, either a wall or a tree or a snake. And we think the whole system is sort of, the brain is processing information sort of simultaneously together in a more rapid and dynamic manner. And we want to really see the whole elephant here. Okay, so what we want to do is identify an entire sensory to motor primary information flow. If one exists in the mouse, there's indications that this is possible in the fly. And so several years ago, we've been thinking about this, but it's always been too hard of a problem. And because most of our pheromone-mediated behaviors, sort of depicted gratuitously by these pictures here, are initiated by a wide variety of sensory cues, not just olfaction. And then they use a wide variety of muscle programs to implicate really dynamic and changing behavior. So, tens to hundreds of muscles that are going off in not exactly a stereotypical or pre-programmed fixed action pattern. And so it's been really hard to be able to get an entire sensory circuit to study this. But several years ago, I had a student in the lab that came up, told me that he had this perfect behavior, which is very simple, and I'm going to show it to you here. Again, let me know in the chat if you can't see the video. But here what we have is a camera on top and a camera below. This is a control odor, and this is the pheromone odor. And as you can see, when we drop the pheromone odor, he started making those little whistling noises, if you can hear them, which are ultrasonic vocalizations, as well as urinating, his urine marks. And just as an understanding of this, so this is a social behavior that can be done with just one animal. And it's really social behavior when an animal, if you have a dog and you've realized as you walk your dog, it's really responsive to the odor environment, sees it as a social cue, and mice do the same thing. And when this male smells a female, smells her pheromones, he wants to advertise his availability to mate. And so he's depositing his own pheromones throughout his territory here, and then using these auditory cues to be able to lure her to have a social interaction. And as you can see, this is odor driven by the external environment, and it's the assay I just showed you. We do over 4 minutes, so 2 minutes with the control odor, 2 minutes with the female odor, and you can see of these 12 individuals here shown along the y-axis, there's a tick made for every urination event, and most of them urinate very rapidly in response to the female odor. And this is ultrasonic vocalizations here, and you can, as you heard in the video, most of them in this blue just start rapidly calling as well. So both of these behaviors are very rapid. They're also not boring, even though they're simple. They're as interesting in their ability to be modulated as more complex behaviors such as fear and mating. And so that is, they're regulated by age. Juveniles don't respond the same way to female odors, and by gender, females respond differently to female odors than males do. They're modulated by experience, so subordinates behave differently than dominants to the female odor. And they're modulated by homeostatic internal state needs, such as whether they're thirsty or sleepy or stressed. And so the other key thing, so it's triggered just by a very simple olfactory cue, so we don't have to worry about the other sensory systems. And these behaviors are released by very simple muscle groups. So for the ultrasonic vocalizations, it's the diaphragm with air pressure, as well as the larynx. And for the urination, it's the bladder pressure. The bladder needs to contract, and the sphincter needs to relax. And in this case, the bladder contraction without the sphincter relaxing doesn't lead to urination. It leads to that sensation of fullness that's very irritating. So really, to be able to urinate, the key thing that your brain does to be able to release urine voluntarily is to relax the sphincter. And we would argue that that's what's occurring here, since the males are not urinating in response to physiological need. Rather, they're voluntarily urinating in response to the female odor. So we set about trying to understand how the brain is initiating these behaviors, and we've made a little progress, which I'll show you in the last few minutes here. Previously, there was very little known about how the brain controls urination. And this set of neurons in Barrington's nucleus, which is a region in the brainstem, had been previously studied and had been implicated in urination. And Barrington's nucleus when it was ablated 30 years ago, was shown to be necessary for urination. But Jason Keller in my lab found a new population of neurons that express estrogen receptor in that area and project to the regions of the brain here, sort of in the middle, in this TGC zone, that are known to project to the urinary sphincter. And if we do fibrophotometry on just these green neurons, we find that we get an increase in fluorescence activity that correlates to these yellow traces here, which are urination events. And if we express channel in those neurons and then drive the behavior with light, here we have an ascending amount of stimulation. That was just 1 hertz, and not really anything happened. If we go to 5 hertz, you can see this animal immediately pees, as shown from the camera down below. We let him move around and increase the stimulation frequency, we can get more robust urination. So these neurons are sufficient to drive urination. And we can also study their role in an anesthetized animal by again, stimulating those neurons with channel in the brain. And then, so here, this animal's anesthetized, the light comes on, and you can see that the urine comes out. So it's a nice behavior that we can study when the animal is anesthetized. If we look again, and this time we're recording from, we're filling with saline, and then recording the pressure, the bladder pressure, as well as an EMG of the muscle. You can see, I'll play it one more time. You can see that as the light comes on, the bladder pressure rises and the muscles start to contract. And so that tells us that all of the elements for urination are being activated. And finally, we can see that it's necessary. So here we're going to start with an inhibitory opsin on these green neurons. We'll start with the light on. And this is just the control odor. We'll drop the female odor here. And this time we just have the camera on top. As the animal moves out, smells the female odor. And you can see there's very little urine came out. It's not robust at all like it was in the control video that I showed you earlier. And now Jason's going to turn off the light. And you might be able to see already there's a little glow underneath. And as he comes out, he's already sort of primed and starts the behavior immediately again. And so, so that tells us that these green neurons are indeed part of this voluntary odor-mediated urine behavior. I'm going to skip that for time. And so those were the neurons that we showed here. They project to the spinal cord and to the urinary sphincter. And there's only about 200 of them in the brain. They're very easy to trace from. And so we've been trying to see if we can make an entire sensory to muscle schematic of the primary transformation. And in green are the nodes that we know are necessary. These grays are hypothetical here, but they're the shortest pathway between what we know about the sensory cues and the motor output. We're hoping that it connects somewhat directly through these nodes or other nodes, that it doesn't sort of go all over the brain. And our goal is to get this primary signal transduction circuit schema in the brain. But I'm going to digress for just a moment more and tell you that as we were validating these nodes that are upstream of those green Barrington's nuclei, as we looked in the LPOA, we saw a different behavior, not necessarily urination behavior. And so that's shown here. And so as you can see, these are those neurons in this LPOA region that we went ahead and expressed channel. And when we turned on the light, the animal didn't urinate at all, but instead was making these ultrasonic vocalization noises. Normally you can't hear these noises. We've slowed it down so you can hear. And this is the sonogram of the noises. And it's quantified here. We found a marker for these neurons. And again, it's this estrogen receptor marker. We don't understand how estrogen receptor is behaving in either of these nuclei. but we're using it as a marker, and it nicely drives both of these behaviors in their different nodes. So we could go ahead and again inhibit those. This time, again, we're in the LPOA, so this does not initiate the urination events, but the calling events. Here we start with the lights off, and this is a male that has arch T in the LPOA neurons with a female, and he's naturally calling to her. Go ahead and turn the light on, and the calls cease right away. But just like with the urinary odors, he still has motivation. He can still sense her. He's interested in her. He just doesn't respond with calling anymore. We went ahead and used a machine learning program called Muppet to be able to look at the types and variety of cues of vocalizations based on their sonogram and frequencies, and find that this artificial stimulation doesn't just produce a sort of stuttering. Instead, it produces an entire repertoire of the male's vocalizations that are most similar to male responding to females. So that female, naturally female. So here's the response to natural females. This is the artificial channel stimulation. And here's a quantification just showing the repertoire and amount of syllables for each type of syllable. And again, it's a wide variety that we get with the channel stimulation. So unlike urination, there's quite a bit known about the circuits that generate vocalizations from work done on song learning in songbirds and on human vocalizations. In the mouse, there's less known, but there's still some. So it's known to be evoked by command neurons in the retroambiguous and dictated by motor neurons in M1 and neurons in the perioctaductal gray, or the PAG. Our neurons are here, an outlier. There's some evidence from some old frog literature that frogs are using an LPOA or MPOA region to drive calling, but nothing has been described in humans, birds, or mice. And so we wanted to know whether the LPOA was projecting directly to the ambiguous or the PAG, or using the known circuits. And both tracing and functional experiments suggest it's plugging into the known circuit. So it's projecting directly to the PAG and stimulating calling through that node. So here is what we found, is that these neurons in the LPOA, these output neurons in the PAG are excitatory. We expected these neurons to also be excitatory, but instead they overlap with inhibitory markers, and fiber photometry of them during natural behavior shows a decrease in fluorescence, so they're presumably inhibitory. And there's a cloud of inhibitory interneurons surrounding the output neurons in the PAG. And if we go ahead and express channel in these neurons, we find that it's enough. So strengthening the inhibitory, presumably inhibitory connections, we find that it's enough to silence calling during natural behavior, and that's quantified here. So we don't think, so we're interested in why use the LPOA to generate this behavior. We don't think that the mice are using ultrasonic vocalizations as a language. Other people have been looking at this for a while, and it may be the case, but there's no indication that they're using a different type of syntax or varying their syllable usage in different environments as of now. But we think that that humans and other animals use a non-language type of vocal communication very effectively. And so this is sort of an innate, a non-structured communication. So in humans, think of it as crying or laughing or shouting. And in animals, whether an animal is purring or growling or barking, you can get a different sense of its behavior. And my student has looked at her cats. So that's her cat making a little meow. Here's her cats being fed, and I hope you can hear that noise is slightly different. And here's her cat getting a bath, and that's almost a little cat howl. And so we wanted to ask, and the difference between these are really the duration of the noise and the amplitude of the noise. And so we wanted to ask, are mouse USVs, do they modulate their duration and volume? And in fact, so we placed a male mouse in a context with a live female odor, anesthetized female, male odor, or an anesthetized male. And we found that the female, he robustly, loudly, and makes loud and long calls to many of the female cues, as shown here. These are different call bouts that have been sorted by length. You can see some of them, these are seconds, some of them are going on for 13 seconds here, and with very dark coloration indicating they're very loud. But with an anesthetized female who has many of the same sensory cues, he's modulated it. So they're very quiet, they're shorter, there's fewer of them. So he is able, it's not as though, sort of like a cricket. Once he makes a noise, it's all or none. They're modulatable. And here what we find is that there's different ability to modulate depending on whether we stimulate from these output neurons in the PAG here, shown here and here, or whether we stimulate from our newly discovered neurons in the LPOA here. So these are the natural variety of cues. And here we find that they're scalable, going from very soft and short to very loud and long. And if we stimulate at very low frequencies in the PAG, they come on very loud. And here they're gated to, they're on the whole much longer. Moreover, we see that if we initiate from the PAG, that those signals are locked to light. They come on right when the light goes on, and they turn off or are silenced right when the light goes off. But if we initiate from the LPOA, we find that they come on a little slower, but most noticeably, they persist long before the light is off. And then sometimes they'll persist quite for many seconds here. So this is 15 seconds. So the decay is over very many seconds after the light is off. And what we found is that the ability to decay is controlled by these intermediate neurons, in that these neurons are locked to light when we turn them on and off. These neurons are locked to light when we turn them on and off, but the calling from these neurons persists. And that's because these neurons, though these inhibitory neurons, relieve the gate right when the light goes off, but they take a while to recover. And this time period here, this is about 5 seconds here. So they can persist, they're asynchronous, and they're persistent in their activity. And we think that's what's generating the behavior. And so overall, what we think is that even when the animal doesn't have its nose in the stimulus, so here, let's see, takes him a moment. Now he's starting to call. But he continues to call even when he's not directly sensing the urine. And we think that's valuable because if he's chasing a female through the environment, if she ducks away for a little bit, that way he still has that sensory evoked desire to call, but it can persist as he loses her immediate sensory information. So overall, what we are doing is trying to get a skeletal information flow that's complete from sensory to motor. We have two different behaviors, very simple social behaviors that have very interesting coding that we'd like to compare and contrast. So we'd really just like to know the information flow. Is it linear feed forward? Is it dynamic? Where is it dynamic? We'd like to mark all the other behavioral events. So I showed you urinating, calling, you saw sniffing and walking, and we're using some machine learning and other tools to try and get a richer description of behavior. We'd like to understand where the features are encoded with these sort of descriptor words that we use, arousal and valence and anticipation. that we think we can study more robustly once we look at the circuit as a whole. And as you're probably most interested, how internal state is modulating and changing the activity patterns, whether the animal is stressed or is tired or thirsty. And we expect that by knowing where to look and looking at the system as a whole, that we'll have a better chance of understanding what each node is doing and when. And so the folks in my lab that did this work were, for the first study with the hormonal modulation, that was first done by a former postdoc in the lab, Sandeepa Day, and is now being looked at the macroscopic level by Nora. And then Jason Keller is now at Josh Dedman's lab, but he's the one that looked at the urination system. And Jingyi has been looking at the vocalization pathway. All these people helped us collaborate with this project. Thank you very much.
Thank you so much, Lisa, for your wonderful talk. So we have some questions. I'm going to go ahead and ask them. So we have the first question from Shital Puttar. Does the male response, urination and USVs, change with female internal state, urine from estrus versus the estrus?
Not at all. Our lab mice, so we don't know. Wild mice, it would make sense that they do, because this is, it's a risky business for them to be leaving cues about where they are for predators, especially hawks have UV vision, so the urination is very risky. So it's a high-risk behavior, but in our lab mice, they'll call to anything that seems like a female, whether it's an estrus or diestrus, they don't care.
Okay, great. How stereotyped is the PLC and odor receptor response to odors between individuals as compared to between internal states or developmental stages?
Yeah. Part of it is control for the individual's internal state, because we think that that is modulating the behaviors. And so as you saw with the qPCR that we did after that PS6 pull down, after we activated neurons, individuals that are sort of in the same internal state, whether they were pregnant or juveniles, they had a very similar response. And so what we imagine, but we haven't shown yet, is that different hormone receptors are modulating, phosphorylating the PLCs and allowing some sensory neurons to be turned on and off differentially in those. So between individuals in the same state, we think that that's very similar. But as you grab individuals out of a cage, they're all in different states. One guy just ate, one guy just got chased around. And so as you get animals that are uncontrolled in different states, we think that they're very different. So we're studying, as our one model internal state, we're looking at stress the most closely, and it's definitely modulatable at that sensory neuron level at the VNO. So it looks like this is probably a good hypothesis, but we're still working on it.
Okay, great. So the next question is, if we maintain continuous estrogen level in the blood of the female, like oral pill, what happens in that case?
Yeah, so we don't. Humans don't have a VNO, so I don't think that we are being modulated in that way. But right, in that case, so if we remove the hormones, they just have a similar constant unchanging response to the male cue. And so we would imagine that if you were a mouse on a birth control pill, that you would have less variation. You were maintaining throughout the days that we were taking the active pill, you would have the same response to the external environment, if that's what I understand. So what we really think is that it is these hormones through these hormone receptors that are on these sensory neurons that are driving the ability to respond to a cue. The sensory receptors are detecting a cue, making it capable, but the hormone receptors are driving the the amount and whether or not that you respond at all. So we were at first concerned that you wouldn't be able to sense a male, but there's also the main nose. So mice have two different sensory chemosensory systems, this vomeronasal system that I've told you about, but also a main nose that they use to sense, like, peanut butter and cookies and regular do associative learning on. And we don't have any evidence that that's being modulated right now. So what we imagine is that she maintains her concept of percepts that are in the environment. Whether there's a male there is probably going through the main nose, but whether she cares about it in a behavioral way that the VNO mediates, that's what's changing.
Okay, so we have two more questions. So first is, how do specific PLCs relate to specific olfactory receptors or other GPCRs? Is there any correlation?
Right now, we haven't sequenced enough neurons just because of the cost. So I'm not going to be able to sequence neurons at this point, the way that the technology is, and address that question specifically. But we've looked at groups of receptors that are in the same receptor family, and we find a bias that the PLCs preferentially bias towards some receptor families and not others. And then we've looked at these two different really large classes of VNO sensory neurons. They're called the V1Rs and the V2Rs. And there's two groups of PLCs that are biased towards one or the other, and then one that's completely unbiased. And that one that was unbiased is the one that in the functional study where we tested the juveniles next to the pregnants, they were on in all of them. So we don't understand those neurons. There's very little understanding of what each type of receptor is detecting, what the ligands are, and what the behavioral significance of it is. And it would be very nice to learn more about that so that we could fill in the why, make more sense of it.
Yeah, I think the next question is also related to, do the different pheromone receptors couple the same type of G protein, GS or Golf?
Yeah, so right now there's thought to be two types of G proteins that correlate with the green cells versus the blue cells, and sort of that crescent that I showed you, if you recall that, the two major kinds of pheromone receptors. It's G alpha I2 and G alpha O. But some of these PLCs aren't known to signal through those two. And so we've just done a search for other G proteins. And again, there's salt and pepper. Some cells have some of these other G proteins, others don't. And so we're looking at how they hook together and whether the pheromone receptors are even signaling through them. There's really good indication now that just the GI2 and the GO are the only primary signal transduction elements. But we don't have a lot of ligands to stimulate. It's the same ligands over and over again. So without being able to stimulate all the other receptor types, it's really hard to test what the primary signal transduction is. So that's now an open question, I think.
Okay, fantastic. Thank you so much, Lisa. So I would like to thank everyone which joined us today. I would like to thank a lot Lisa for joining us and telling us about her work. I hope to see you all next week, and I hope to see whoever joined for the Meet the Speaker session, which will happen now on Zoom. So see you all now. See you there, Lisa.
Thank you. Thank you for the talk. I really you guys staying up. I know you for this regular time. Very nice. Visiting.